Today, we're going to talk about something that sounds like it's straight out of a movie. A technology that gives us the power to rewrite life itself. A tool that can, with incredible precision, edit the fundamental genetic code of pretty much any living thing. And no, this isn't science fiction anymore. It's the science of CRISPR.
I mean, really think about that for a second. It's a huge question, and it's one we now have to take seriously. This technology has thrown open a door to possibilities that were, frankly, unthinkable just a couple of decades ago, from wiping out genetic diseases to completely re-engineering the world around us.
But to really get a handle on this powerful future, we've got to go back in time. It all started with the discovery that, at first, seemed like nothing more than a weird biological quirk, a strange little puzzle hidden away in the DNA of bacteria.
So the story kicks off way back in 1987. Researchers in Japan are looking at E. coli DNA, and they find these bizarre repeating segments. For years, nobody had a clue what they were for. Then, around the mid-2000s, there's this huge breakthrough. Scientists realize, wait a second, this isn't just junk DNA, it's a defense system. And that discovery set the stage for 2012, the year biology would change forever.
They gave this puzzle a name that's, well, it's a bit of a mouthful, but it perfectly describes what they saw. CRISPR. It stands for Clustered, Regularly Interspaced Short Palindromic Repeats. I know, but it's really just a very literal, very scientific description of these weird patterns.
Now, that P in CRISPR, for palindromic, turned out to be a really important clue. A palindrome is something that reads the same forwards and backwards, like the word race car. This kind of symmetry in DNA usually means it's doing something very, very specific. So, what was the big secret? What was this genetic puzzle all about? Well, the answer was absolutely mind-blowing. These repeating bits of code weren't random at all. They were the backbone of an ancient and incredibly sophisticated defense system.
Yeah, you heard that right. CRISPR is how bacteria fight off infections. Think about how our own immune system works. If you get sick, your body learns to recognize the bug and remembers how to fight it off next time. Well, it turns out, tiny little bacteria have been doing a version of that for millions and millions of years. You see, you gotta picture it like this. Bacteria are in a constant, epic war with viruses called bacteriophages. These viruses are nasty. They latch on, inject their own DNA, and turn the bacterium into a virus-making factory. It is a microscopic battle for survival, happening all the time.
So, if a bacterium manages to survive one of these attacks, it does something brilliant. It literally snips out a piece of the invader's DNA and weaves it into its own genome, right there in those CRISPR spots. It's basically taking a genetic mugshot of the enemy.
It's a living genetic memory, a library of past attackers. Every time it survives a new virus, it adds another mugshot to the collection, passing that knowledge down to all its descendants. It's incredible.
Okay, so this natural defense system is already super cool, right? But the real revolution, the part that changed everything, started when scientists figured out how to take this ancient bacterial weapon and turn it into a tool that we could control.
So how does the bacterium use its mugshot collection? Well, it makes little RNA copies of those stored viral snippets. These RNA copies then act as guides, and they team up with a protein called Cas9. Now, you should think of Cas9 as a pair of molecular scissors. The guide RNA leads the Cas9 scissors right to any invading virus DNA that matches the mugshot, and snip, the virus is neutralized.
And this was the big aha moment. Researchers Jennifer Doudna and Emmanuel Charpentier had this incredible thought. If a bacterium can program this system to find and cut viral DNA, then maybe, just maybe, we could reprogram it to find and cut any DNA sequence we want in any living thing.
And here's the genius hack that made CRISPR a tool for everyone. See, in nature, the system is a little complicated. It uses two different RNA molecules. But in the lab, scientists figured out how to fuse them together into one simple, elegant molecule, the single guide RNA, or sgRNA. This one little tweak made the whole system unbelievably easy to use.
So this single guide RNA is the programmable part. It's like the GPS for our molecular scissors. Scientists can design a guide to match basically any stretch of DNA they want to target. You just cook up the right guide, mix it with the Cas9 scissors, and you've got yourself a precision-guided gene editor. It's that straightforward.
All right, so we've got our guide and we've got our scissors. But how does this system actually go from cutting DNA to rewriting the genetic code? Let's walk through it.
It's really a beautiful four-step process. First, the guide RNA and the Cas9 protein link up. Second, this little complex gets inside a cell and just starts scanning, zipping along the DNA looking for a match. Third, when it finds the exact DNA sequence that matches its guide, it locks on tight. And finally, bam! The Cas9 scissors do their thing, making a clean cut right through the DNA.
But see, making the cut is only half the battle. The real magic, the part where the editing happens, is what the cell does next to repair that break, and it's got a couple of options.
The cell basically has two main repair pathways. The first one is kind of the fast and messy option. The cell just tries to glue the two broken ends back together as quickly as possible. This can be a little sloppy and often introduces errors, which is actually useful if you want to knock out or disable a gene. But then there's the second, more precise path. If scientists also provide a new, healthy piece of DNA as a template, the cell can use that to make a perfect repair, letting us rewrite the genetic code or even slot in a whole new gene.
And this ability, the power to easily turn genes off or rewrite them with pinpoint accuracy, has kicked off nothing short of a revolution in biology and medicine. It's a total game changer.
I mean, the possibilities are just staggering. We're talking about taking a patient's own immune cells, engineering them to hunt down cancer and putting them back in, developing super fast tests for viruses, creating crops that can survive droughts, and, for the first time ever, we have a real shot at curing thousands of inherited diseases like sickle cell anemia and cystic fibrosis.
Just look at one example, HIV. The reason it's so tough to cure is that the virus literally hides its DNA inside our own cells. But with CRISPR, the strategy is totally different. We can send in those molecular scissors on a search and destroy mission to find that hidden viral code and just cut it out for good.
But, and this is a huge but, this incredible power comes with some profound, heavy questions. The same tool that could cure inherited diseases could also be used to edit the genes of human embryos, making changes that would be passed down for generations. We've literally built a tool that lets us direct our own evolution.
And that really leaves us with the ultimate question. Now that we have this almost unimaginable power, how do we choose to use it? Where do we draw the line between healing and enhancement? Between curing disease and fundamentally rewriting what it means to be human? This isn't just a question for scientists anymore. It's a conversation for every single one of us.